students, welcome to Life Sciences with Jyotishri. So in this module, we will be learning about the basics of molecular biology. So in a series of sessions, we will be discussing about basis of molecular biology. So there are a number of processes necessary for the continuation of generation, growth and development of organisms called fundamental process. These includes your replication of genetic material, damage and repair and recombination in genetic material, isn't it? So all these mechanisms, that is your replication, transcription, translation, gene expression, everything you will be learning in molecular biology. So in this module, we will be discussing about the basics of molecular biology. So what is molecular biology? Molecular biology is nothing but the study of macromolecules at its molecular level. So macromolecules are nothing but your DNA, RNA and as well as your protein molecules. Protein molecules are nothing but your biomolecules, but your DNA and RNA makes up your macromolecule, which will help your protein molecule to be get expressed for a specific character, right? So coming to the definition of molecular biology is a branch of biology that concerns with the molecular basis of biological activity in between cells and including molecular synthesis, modification, mechanisms and interactions. So that we have already discussed that the study of macromolecules at its molecular level. So the whole thing can be explained with your central dogma. Central dogma is nothing but it is the flow of information from DNA to your protein molecule, and in between you will come across several what do you say, mechanism, events of mechanisms like your synthesis of your DNA molecule as well as your RNA and your protein molecules, that is your replication, transcription and translation. So this molecular biology includes your biology, chemistry and also interactions between your DNA, RNA and as well as your protein molecules that we have discussed. Okay, now let us see. These are the components involved in molecular biology, that is your your DNA, RNA, and as well as your protein molecule. So this is solely carried out with your gene, that is nothing but your unit of heredity, isn't it? So the DNA segments that carries genetic information are called genes, and it is normally a stretch of DNA that codes for the type of protein or a RNA chain that has a function in the organism. Genes hold the information to build and maintain an organism's cells and pass genetic traits to offsprings. So gene is the unit of hereditary, and these genes are carried by your chromosomes, isn't it? So these chromosomes carries your your genes, and these genes carry your DNA molecule from one generation to another generation. Chromosomes mainly consist of nucleoproteins having two components, that is your nucleic acids and proteins. One of these should obviously constitute the genetic material, and that genetic material is nothing but your nucleic acids. So what are these nucleic acids? How are they made? So these nucleic acids are nothing but they are made of your nitrogen bases, and there are your adenine, guanine, cytosine, thymine and as well as your uracil. So these nitrogen bases are made under two categories, that is your purines and pyrimidines. So purines is having a heterocyclic ring with attached with a benzene ring, and whereas your pyrimidines, these are not your pyramids but pyrimidines, these are only having a benzene ring. So under your purines, adenine and guanine are present, and under your pyrimidines, cytosine, thymine and as well as your uracil molecules are present, that are your nitrogen bases. And coming to the pairing of your DNA molecule, so in your DNA molecule, adenine, thymine, guanine and cytosine are present. So adenine is always double bonded with thymine and your guanine is always triple bonded with cytosine. So in place of your RNA, in place of your thymine in RNA, uracil is present. So adenine is double bonded with uracil. So it's a hydrogen bonding. Okay. So this is your sugar base and your sugar and your nitrogen base. So your yellow color is your sugar molecule, that is your ribose sugar, and blue indicates your nitrogen base. So sugar, combination of a nitrogen base with a sugar is nothing but your nucleoside, and the combination of your sugar as well as your nitrogen base and your phosphate group makes your nucleotide. You can easily remember all these components that nucleotide makes a tide, T-I-D-E, tide, nucleotide. T means indicates three. Okay, three. It's a three component molecule which it is having three components, that is your ribose sugar and as well as your nitrogen base and with your phosphate group. Nucleoside, SIDE, side means two, second. Two, only two. That is your ribose sugar and as well as your nitrogen base. Okay, so let us discuss about your DNA molecule. So DNA is a nucleic acid that contains the genetic instructions used in the development and functioning of all non-living organisms and as well as your viruses. Got it? So what's this DNA? So this DNA is nothing but this was first demonstrated by Watson and Crick in the year 1953, and they has stated that this is a double helical structure with two polynucleotide chains connected by nitrogen bonds and running in opposite directions. Okay, so this is your DNA molecule running in opposite directions, and there are several postulates related to the structure of your DNA molecule, that the number of adenine molecules always equals to the number of thymine molecules and the number of cytosine molecules always equals to the number of guanine molecules. So this is given by a rule that is nothing but your Chargaff's rule. Okay, which states that there is no restriction in the ratio, that is A plus T is by your cytosine to the guanine. So that is nothing but your Chargaff's rule. Yes. Okay, now coming to the size and the length and the number of base pairs per turn is given here. So generally there are different types of DNA, A DNA, B DNA, C and as well as your Z DNA, and each turn, the length of each turn is 34 angstroms, and each turn, and in each turn there are 10 base pairs, that is your adenine, guanine, whatever it may be. So it is a double bonded, it is having a complementary strand base pairing with the opposite strand, isn't it? So that is how your DNA molecule is formed, and each turn 10 base pairs are present, and the length of a turn is 34 angstroms, and the length in between two base pairs is 3.4 angstroms, and coming to the diameter of your DNA molecule is 20 angstroms. It varies according to the different types of DNA. Generally we will be discussing about your bDNA. Okay, so this is how your adenine is double bonded with thymine and guanine is triple bonded with cytosine. And one more thing, what about the sugar? So the sugar molecule that is present in your DNA is deoxyribose sugar, and whereas in your RNA it is only ribose form. At the second position, the oxygen is removed, and hence it is deoxy form in your DNA molecule, and that is the main difference between your DNA and as well as your RNA molecule. So this is your RNA. RNA is biologically important type that consists of long chain of nucleotides, and it is also consists of nitrogenous base, ribose sugar and your phosphate group. So this is double stranded RNA. In some cases, in some exceptional cases, it's only a single stranded RNA, but it folds upon itself to make it to show its structural configuration as a double bonded structure. Okay. So there are the different types of your RNA molecule: messenger RNA, ribosomal and transfer RNA. Messenger RNA codes for your protein in all organisms. rRNA functions, it shows its function during translation, that is your protein synthesis, and transfer RNA also has its own function in protein synthesis, but it carries your activated amino acid to the site of your protein synthesis, that is nothing but your ribose. Okay, and these are the post-transcription modifications: telomerase RNA and yRNA and antisense RNA. So we will be discussing all these things, all these your types of RNA, antisense RNA, post-transcription modifications in the coming series of sessions. So this is messenger RNA, which codes for your nucleotides, and this is having both your introns and as well as your exons. Okay, so introns are removed and only the presence of exons will give up your mature RNA molecule. And this is your transfer RNA, which consists of 5 arms, and all these things are generally used for your carrying of your activated amino acid to the site of your protein synthesis. So that is the function of your tRNA, D arm, T comma psi arm, and your anticodon arm. Okay, so these are the different arms that are present in your RNA molecule. Next, rest of them are also present. They are also having its own functions for carrying of your activated amino acid to the site of protein synthesis. So this is ribosomal RNA, and in eukaryotes the sedimentation, the type of RNA is ATS type, as indicates so sedimentation coefficient, and this ATS type is having again two units, smaller unit and your larger fragment and smaller fragment. Larger fragment is 60S type and the smaller fragment is 40S type, whereas in your prokaryotes it is 70S type, that is sedimentation coefficient, and 50S is your larger fragment and 30S is your smaller fragment. Coming to the difference between your RNA and as well as your DNA molecule, RNA nucleotides contains ribose sugar, and we have already discussed in DNA deoxyribose form is present, and RNA has a base, that is sugar, uracil, and in place of uracil in DNA molecule thymine is present. So as we have said that at the second position, the hydroxyl group lacks oxygen and hence it is deoxy form in DNA, and ribose form is only ribose sugar in your RNA molecule. RNA is usually single stranded and DNA is double stranded. Okay, so these are your proteins. The third component of your molecular biology, macromolecules. Proteins are also known as polypeptides, and they are made of amino acids arranged in a linear chain and folded in a globular form. The sequence of amino acids in a protein is defined by the sequence of gene which is coded in genetic code, and your genetic code is always triplet in nature. That is how the language of DNA and from your RNA and the language of protein molecule is made, and your gene expression is observed. So the whole thing is carried out, that is nothing but your central dogma. So the basic players in the molecular biology are your DNA, RNA and protein, and these three are observed in the playground of your central dogma. So this is one-sided flow of genetic information, that is your synthesis of your DNA molecule from another DNA molecule, that is replication, and synthesis of your RNA molecule with the help of your DNA, that is transcription, and synthesis of your protein molecule with the help of your RNA, that is translation. So in the coming series of sessions, we will be discussing about the mechanism of replication and the enzymes involved in replication, and as well as proteins and as well as the inhibitors of replication. And next we will be learning about mechanism of transcription, same thing, post transcription modifications with your RNA molecule, and as well as the synthesis of protein molecules, that is translation. Thank you.